glycine is the simplest amino acid, right? And from glycine metabolism, we will get an idea about the basic information about glycine, its synthesis and degradation, and the specialized metabolites produced from glycine metabolism. Okay, so glycine metabolism, though glycine is a non-essential amino acid, but glycine is involved in production of various important biological compounds. So glycine metabolism is important, and there will be few metabolic disorders related to glycine metabolism. Now see, what is glycine? Glycine is the simplest amino acid. We can see amino acid structure with no extra side group. The R group here is hydrogen. So that makes the glycine the simplest and the smallest amino acid. And what else property is there? Because of the absence of this R group, presence of H. So two H's are there, two hydrogens are there. So there is no chirality. So chiral carbon is not present in the single only amino acid, that is glycine. Glycine is optically inactive. No chiral carbon in glycine, that's why it is optically inert or optically inactive. The mirror images will be superimposable, right? Is it clear to everyone? It is a simplest non-essential amino acid with optically inactive carbon, and it is glycogenic amino acid. That means in further metabolites will give rise to glucose. Glucose will be produced from the last metabolite, and the biologically important compounds that are produced from glycine metabolism, they are heme, purines, creatine, glutathione, etc. and many more. Okay, and glycine is an important part of our collagen protein. Okay, so every third amino acid in collagen is glycine. That makes the 30% of collagen is only of glycine. Clear? Now this is just an depiction what are formed from glycine: heme, glutathione, creatine, purines, that will further lead to formation of nucleic acids. Bile acids, there is conjugation of glycine with bile acids, and it will form the conjugated bile acids. Okay, and for detoxification, yesterday we saw in the treatment of urea cycle disorder, benzoate were used for glycine elimination, so that is for detoxification. Now the synthesis and catabolism of glycine. These three pathways are very important in glycine synthesis and catabolism, because these three pathways are bidirectional. Okay, so they can form glycine, and glycine can be catabolized into these three components. So first of all is serine to glycine. Serine is another non-essential amino acid that can be produced from glycine, and so glycine is also non-essential because it can be produced in our body from serine. Okay, so serine to glycine and glycine to serine is one of the major synthesis and catabolism process of glycine. Next is oxidative cleavage of glycine. If you remember the structure of glycine, it is made up of OH group and amino group and a carbon group. Okay, so ultimately it will lead to formation of carbon dioxide and ammonia. Okay, so this breakdown, this is mediated by glycine synthase enzyme, and that is a multi-functional enzyme. Okay, so and vice versa, carbon dioxide and ammonia can bind together to form glycine in presence of that same enzyme. Okay, and the last thing is oxidative deamination of glycine will give result to glyoxylic acid. Okay, why glyoxylic acid is important? Because this will in turn give rise to oxalates, and oxalate crystals are very important in case of stone formation in our body. Okay, so thus if there is increased glycine intake, and the metabolism will lead to production of glyoxylate or glyoxylic acid in turn, which will give rise to more oxalate stones. Okay, and this is also bidirectional reaction. Now coming to individual reactions, the synthesis of glycine. Here we can see serine is converted to glycine by serine hydroxymethyl transferase. Here the structure of serine is given. Okay, so what is the difference? There is an extra methyl group or hydroxymethyl group in serine. Serine is an OH group containing amino acid. So the hydroxymethyl group, if transferred to something else, it will form glycine. Clear? That's why here we use a cofactor, that is tetrahydrofolate. Tetrahydrofolate in presence of pyridoxal phosphate. Okay, pyridoxal phosphate in presence, this is a coenzyme, and the enzyme is serine hydroxymethyl transferase. Serine, from serine the hydroxymethyl group will be transferred to tetrahydrofolate. Is it clear? Clear to everyone? So this reaction will form glycine, and this is backward reversible reaction. Okay, so if needed, serine can also be formed from glycine by the same enzyme and coenzyme and cofactor. Okay, now moving to the next amino acid that also gives rise to glycine, that is threonine. Okay, so threonine, the structure of threonine is this. So here this part is extra, this acetyl group is extra. Clear? So if we break down threonine, we will get glycine and acetaldehyde. So this reaction is facilitated by threonine aldolase. Clear? So two amino acids we get from where glycine is produced. One is serine, another is threonine. Next is the reaction where glycine can be formed from carbon dioxide and ammonia by glycine synthase enzyme complex. It is a multi-enzyme complex. Okay, this reaction occurs in various steps, but for a depiction, easy depiction, this is shown as this. The glycine synthase needs oxidative cleavage, okay, or oxidative addition. So here NADH is used, and for the transfer of amino group, the tetrahydrofolate, or the methylene group, for transfer of methylene group, tetrahydrofolate acts as the cofactor. Okay, so if asked about glycine synthase, it is a multi-enzyme complex which needs NAD for oxidative reaction, and cofactor here is tetrahydrofolate, and the coenzyme is pyridoxal phosphate. Okay, and the reverse reaction can also take place from glycine, carbon dioxide and ammonia can be formed. Is it clear? বোঝা গেছে এই পর্যন্ত? Next, for further metabolism, serine can produce pyruvate by serine hydratase enzyme. Okay, so the metabolic fate of glycine in energy pathway will be production of pyruvate and thus production of glucose. Okay, that's why glycine is glucogenic. The formation of glucose from glycine is via serine. This reaction from serine, decarboxylation, removal of carbon dioxide, decarboxylation reaction of serine will give rise to ethanolamine, and this ethanolamine in further reaction will give rise to choline. Okay, the neurotransmitter choline, and glycolate also is another substance from which we will get the glyoxylate. Okay, so the glyoxylate compound can be formed from serine or directly from glycine. If it is forming from glycine, the enzyme here is glycine transaminase, and in this case also pyridoxal phosphate is the coenzyme. Okay, and from glyoxylate to glycine formation is by transamination reaction, whereas glycine to glyoxylate is by glycine oxidase. Okay, all these reactions, these steps are not important, like you need not to remember the details of the step, but the formation you should remember. Okay, that glycine can be converted into glyoxylate and glyoxylate can be converted to glycine. Okay, further here glyoxylate, I was telling you glyoxylate will form oxalate. Okay, and that oxalate if increased, it can form oxalate stones. It will combine with calcium and will form calcium oxalate stones, and the oxalate part is removed by urine or excreted by urine, and the other part or the formate part, it is one carbon compound that is provided by glycine. So one carbon metabolism is another important part of amino acid metabolism. There are multiple one carbon group that can be donated from individual amino acid metabolism to some another compound. Okay, so here what is happening, the formate that is coming from glycine via glyoxylate will be transferred to tetrahydrofolate to form N-formal tetrahydrofolate. So that will further be required in some other reactions. Okay. Okay. Now coming to synthesis of individual product formation from glycine metabolism, and I will not go in details in first few things because that will be taught in details in the individual subject class. Okay, so here it is the formation of purine ring that will be taught in your nucleotide class. Okay, nucleotide chemistry class. So you just have to know that synthesis of purine ring, here it is a purine ring and the carbon number 4, 5 and N7, this part is coming from glycine. So see, glycine has 2 carbon 1 nitrogen. So 2 carbon 1 nitrogen. So whole of the glycine is incorporated in purine ring. That is why this comes first. The metabolic product of glycine or the special products from glycine, that is purine ring. Okay, as the whole of the glycine is being incorporated. Second is synthesis of heme. Porano hoye geche to heme synthesis. So the first step of the heme synthesis is succinyl-CoA with glycine in presence of ALA synthase will give rise to ALA. Okay. So this is also has been taught to you in details. Now neurotransmitter, as I said that glycine can form choline and so on. So it along with taurine, it forms an inhibitory neurotransmitter, and that glycine acts by increasing the permeability of chloride ions. Okay, so that is the mechanism of action as neurotransmitter, and it is inhibitory in nature. That's why the metabolism of glycine is important in case of any neurological normality. Okay. Now coming to the most important thing, that is glutathione. Glutathione is a tripeptide. What is tripeptide? Made up of 3 amino acids. Okay, 3 amino acids, glycine, cysteine and glutamic acid. Okay, so it is written as GCG, but the cysteine stays in dimer form. যখন আমি বলেছিলাম যে সাইড চেইনস অফ অ্যামাইনো অ্যাসিডস অর স্পেশাল বন্ডস ফর্ম, দে ক্যান ফর্ম স্পেশাল বন্ডস, তো সিস্টিন ক্যান ফর্ম disulfide bond. Okay, and by disulfide bond it will stay in dimer form as cysteine. Okay, so this cysteine generally stays as cysteine. Okay, so 2 cysteine molecules will be there by SH-SH bond. Okay, so glutathione is written as this also and also as G-SH-SH-G. Okay, so I have forgot to write that, but you please note that glutathione can be depicted as G-SH-SH-G. Okay, SH-SH is to denote the cysteine, the double bond sulfide bond from cysteine. Okay, now see, this glutathione is very important because it is a component of antioxidant enzyme glutathione peroxidase. Okay, which is used in free radical scavenging, or it acts as the antioxidant. Okay, so we will learn in details in our free radical classes, okay, regarding glutathione, but for now you just know that it is a part of glutathione peroxidase enzyme complex and it is used for free radical scavenging. Now the main function of it is to maintain integrity of RBC membrane. The glutathione covers the RBC membrane and it acts as the hydrogen donor. Okay, in case of antioxidants, what happened? The superoxide is there. So the superoxide, that is highly oxidative. Okay, it will oxidize the molecules, but the glutathione that stays in the reduced state will give hydrogen ion to the superoxides and the negative charge will be balanced. And so it is used for detoxification and it protects hemoglobin from oxidation, and it also helps in transport of amino acids by Mr. cycle, and it also acts for some enzymes as cofactor. Okay. Now the next thing, that is conjugation. Conjugation of glycine in where it is used in formation of bile acids. Okay, so glycine will bind with cholic acid to form glycocholic acid, and quinodeoxycholic acid binds with glycine to form glycoquinodeoxycholic acid. These are the major primary bile acids. Now come to the formation of collagen. So collagen is major matrix protein. Collagen is the major matrix protein in our body, and see here 3 amino acids are repeated in sequence, right? So what are the 3 amino acids? Glycine, proline and lysine, and the hydroxyproline and hydroxylysine is also there. Okay, so proline can be hydroxylated, but the fixed third amino acid is glycine, and this is a single strand of collagen and it stays in a triple helix manner. One collagen molecule is composed of 3 such peptide bonds, peptide chains. Okay, so 3 peptide chains stays in a triple helix manner to give rise to a single collagen chain or single collagen molecule, and the collagen fiber looks like this. So individual these threads, they are composed of triple helix structure. Is it clear? Now see, so about 30% of collagen is composed of glycine, and as the glycine is smallest and the non-polar amino acid, it can fit into the helices, okay, in the inner part of the helices. So that's why it is the most important amino acid in collagen. Okay, now detoxification, you already know that detoxification of glycine is done by benzoate and it is excreted in the form of hippuric acid. So in case of ammonia toxicity, benzoate can be used for removal of nitrogen compounds in form of hippuric acid as it binds with glycine. So glycine here acts as detoxification agent also. Next, glycine can synthesize glucose and it is constituent of various tissue proteins and part of enzyme systems and hormones also. Okay, so these are the special products that are formed from glycine. So just for a quick recap, what are the special components that were formed from glycine? First of all, purine. The C4, C5 and N7 part of any purine ring is coming from glycine. Next, the synthesis of heme, the initial step starts with glycine and succinyl-CoA. And third part is it can act as neurotransmitter also, inhibitory neurotransmitter with taurine. Okay, and next, that is formation of glutathione. So how glutathione is formed? It is a tripeptide. There is glycine, cysteine and glutamic acid. Right? So glutamic acid, or that is glutamate. Okay, so glycine, cysteine and glutamate, but the cysteine stays as dimer form, that is cysteine. Okay, so it is depicted at GSHSHG. Okay, and it can stay in oxidized or reduced form. If reduced form, that will be SH. If oxidized, it will be S double bond S. Okay. So the H that will be donated and it will be oxidized, and that H or the reduced form is used to nullify the superoxide ions. Okay, superoxide or the superoxide, the components that are free radicals. Free radicals are nullified by the hydrogen from glutathione, and glutathione covers the RBC membrane to protect it from all the free radicals, and it prevents hemoglobin also to become oxidized, and it acts as carrier for amino acids in few cases in Neisser cycle, and it can act as cofactors in various enzyme reactions. Clear? Next, that was conjugation of glycine to form primary bile acids. So what were they? Glycine with cholic acid will form glycocholic, and glycine plus canodioxycholic will form glycocanodioxycholic. Okay. Next is collagen formation. As we can see that every third amino acid in collagen is glycine. It contains compose the 30% of collagen molecule. Okay, and it can and the detoxification is also very important. If there is ammonemia, so we can use benzoic acid or benzoate to eliminate the glycine and to reduce the nitrogen load, and last but not least, it will synthesis glucose and will take part in formation of various other enzymes, hormones and proteins. Now another important product that is formed from glycine is creatine, but creatine is also made up of two other important amino acids. They are arginine and methionine, GAM. Creatine or creatinine, they are formed from glycine, arginine and methionine. So this formation of creatine occurs in three different places. The first part is where glycine binds with arginine and the guanido group is transferred by amidinotransferase enzyme, and it forms guanido acetate. Okay, guanido acetate, and ornithine is released. So the first reaction takes place in kidney. Formation, in case of formation of creatine, the first reaction where glycine binds with arginine. Glycine binds with arginine, where the guanido group from arginine is transferred to glycine to form guanido acetate, and the enzyme is amidinotransferase, and the ornithine can go back to urea cycle to form arginine again. Okay. So arginine will transfer guanido group to glycine by amidinotransferase enzyme to form guanido acetate in kidney, and ornithine will be released for urea cycle. Okay, this guanido acetate travels to liver, and there what happens, a methyl group is transferred, a methyl group is added to this. In guanido acetate, a methyl group from S-adenosyl methionine, here the methionine comes, taito. So glycine, arginine and methionine will form creatine, and here in liver guanido acetate joins with a methyl group in presence of methyl transferase enzyme. Methyl transferase enzyme helps in transfer of methyl group from S-adenosyl methionine to guanido acetate, and it will form creatine, and S-adenosyl homocysteine will be released. Okay. So this is a very important amino acid metabolite from methionine, which readily gives methyl group. It acts as a general methyl group donor in various reactions. So remember this, SAM or S-adenosyl methionine, that gives the methyl group. SAM is a general methyl donor, methyl group donor, and after donating methyl group it will form S-adenosyl homocysteine, and the creatine is formed here in liver. Okay, so the methyl transferase enzyme is in liver, that's why the formation of creatine is occurring in liver. Is it clear until this? So the first part was in kidney and the second part is in liver. Now this creatine that is formed in liver now is transported to muscle. Creatine kothay thake? Muscle e thake. In all our muscles there is creatine, and what is the role of creatine? It acts as energy currency, ATP. How? See, what happens in muscle, the creatine, or in the transport pathway from liver to muscle, it is converted into creatine phosphate by ATP molecule. A phosphate is added to creatine by ATP, and this is facilitated by creatine kinase. Creatine kinase or creatine phosphokinase enzyme. That's why creatine kinase is very important in clinical diagnosis, all the clinical diagnosis. So here what happens, this is a reversible reaction, bidirectional reaction. When needed, creatine can form creatine phosphate, and when there is requirement of energy in muscle tissue, muscle cells, so creatine phosphate will release ATP and will be converted into creatine again. So there is no need of ATP itself in the muscle. It is needed, but if not present, creatine phosphate can give the phosphate, and these phosphate bonds are high energy phosphate bonds. Right? So it is consuming an ATP to form creatine phosphate, and if required, an ATP can be released by joining an ADP, and creatine will be formed back. Okay. So is this clear? So this cycle from creatine to creatine phosphate and creatine phosphate to creatine in muscle, this takes place simultaneously all the time. Okay, and this is known as Lohmann's reaction. So Lohmann's reaction is nothing but the conversion of creatine to creatine phosphate and the reverse step. Okay. Now see, what is creatinine? Until now we formed creatine and creatine phosphate, but what is creatinine? Creatinine is nothing but the anhydride form of creatine. Anhydride form of creatine, that is removal of one molecule from creatine will give rise to creatinine. Creatinine is nothing but anhydride form or dehydrated version of creatine, and this is excreted in our urine, and it can also be formed from creatine phosphate by removal of an inorganic phosphate. So creatinine can be formed from creatine by removal of one molecule of water, or from creatine phosphate by removal of one phosphate, inorganic phosphate. Clear? And this creatinine, this is inert substance. This cannot be reabsorbed, or this cannot be produced in our renal tubular cells. Okay, so that's why this is freely excreted in our urine, and this makes creatinine very important. Okay, why creatinine is very important? Because it is metabolically inert and it is not reabsorbed or not produced in the renal tubular cells. Okay, that's why we use serum creatinine for estimation of kidney function test. So whether the kidney is functioning, a normal kidney will never reabsorb or produce the creatinine. In case of creatinine excretion, the amount of creatinine excreted, this is fixed. Okay, daily excretion of creatinine is fixed, and this depends on the body mass, muscle mass. So it is equivalent to the creatine or creatine phosphate we have in our body, and the creatine we have in our body is equivalent to the muscle mass we have or the muscle protein we have. Is it clear? So creatine is converted into creatine phosphate, and that is stored in our muscles. in equivalent amount that is required for our muscle mass. So the excretion of creatinine is from the breakdown of creatine or creatine phosphate. So this represents, creatine excretion represents our muscle mass, okay, muscle mass or the volume of our muscle. And see, this is also very important, normal concentration of creatinine in human, this is different for men and women because body mass in men is males are more, so that's why creatinine is also more in men, and in women it is this. And urinary creatinine excretion, that is 1 to 2 gram per day, okay. And the increased output of creatine, okay, so generally creatine is not excreted, creatinine is excreted. So increased output of creatine can be seen in muscular dystrophy, diabetes, hypothyroidism and starvation. In case of diabetes, what happens, there is loss of kidney function, that's why it will be excreted more. But in case of muscular dystrophy, hypothyroidism and starvation, what happens, there is more muscle breakdown. Muscle breakdown is increased, that why increased creatine production is there and it will be excreted by our urine. Is this clear? Clinical significance of creatinine is clear though? Now coming to metabolic disorders of glycine. So glycine can cause glycinuria. If it is excreted in increased amount in our urine, it is known as glycinuria. This is an inborn error of metabolism and can be detected in childhood only. And in this case, glycinuria, plasma concentration of glycine can be normal, okay, but glycine will be excreted much in urine, and there is a defect in renal tubular reabsorption. So glycine, if excreted in our normal urine, it can further be reabsorbed from our tubular cells or tubules, okay, renal tubules. But in case of glycinuria, there is defective renal tubular reabsorption, and there is a tendency to form oxalate renal stones, okay, in this case. Glycinuria. In glycinuria, what happens, the absorption, reabsorption is hampered and it can form oxalate stones in kidney only. And oxalate stone in human body comes from, 60% comes from glycine and ethanolamine. Ethanolamine was coming from? Ethanolamine was coming from serine. Decarboxylation or removal of carbon dioxide from serine will give rise to ethanolamine. And 30% of oxalate stone, they come from vitamin C, okay. So major component is glycine and serine, 30% comes from vitamin C, 10% is from the oxalate rich foods. Disease is primary hyperoxaluria, okay, oxaluria, primary hyperoxaluria, that is this pathway from glyoxylate to glycine formation, and this was mediated by which enzyme? Glycine transaminase, okay. So the transaminase enzyme is defective or deficient in this case, that's why glyoxylate cannot form glycine, so it will be directed to oxalate pathway. You remember from the main flow chart where glycine was converted to glyoxylate and glyoxylate was forming oxalate and formate. Right? So here glyoxylate will form oxalate and this oxalate will bind with calcium and will cause renal stones, okay, stones in urinary tract and in kidney. So there is a chance of recurrent infection or recurrent UTI in case of hyperoxaluria, and obviously there is presence of stones in kidney, and if detected early it is good, or it is also an inborn error, okay, autosomal recessive type of inborn error. So it mostly detected in childhood, in infancy only, but if this becomes severe, so death can also occur, okay. Okay, another disorder from glycine metabolism, that is non-ketotic hyperglycinemia. Hyperglycinemia, where there is defect in glycine cleavage system. For the catabolism of glycine that was occurring, or glycine that was converting into carbon dioxide or nitrogen, or the end step, or the catabolism of glycine or breakdown of glycine that was occurring by enzyme glycine synthase, it is defective. When the glycine synthase enzyme is defective, it will lead to hyperglycinemia or increased serum level of glycine. Okay, so this enzyme is defective here, so glycine cannot be converted into carbon dioxide and ammonia, and it will lead to increased serum glycine. Here severe mental retardation is seen, and death in very early childhood is seen, and if not, there is neurological defects. Okay. Now to summarize the glycine metabolism. For synthesis, glycine is synthesized from serine, threonine, glyoxylate, and from carbon dioxide itself. Okay. For degradation, the same pathways, but in case of threonine, it will be oxalate. The formation of oxalate. Serine can be formed from glycine, glyoxylate can be formed from glycine, and from glyoxylate, oxalate will be formed, and glycine can be broken down to carbon dioxide and ammonia. Now the metabolic role or the special products that are formed from glycine, it is heme, creatine, glutathione, purines, detoxification purpose and conjugation of bile acids. Formate is one carbon compound, that is the one carbon metabolism is also facilitated by glycine metabolism. Collagen formation, and glycine acts as neurotransmitter also, and in case of metabolic disorder we may see glycinuria, primary hyperoxaluria, and non-ketotic hyperglycinemia. Clear? Is it clear to everyone? Okay. Now very quick MCQs. Glycine is used for synthesis of all the compounds except pyrimidine ring. In the body, glycine is used for synthesis of all except what is guanine? So what is the answer here? Glutamine. Okay. Next, normal serum creatinine level is 0.7 to 1.4. Very good. Creatine is synthesized from the amino acids except aspartic acid. Good. And next is source of oxalic acid in urine. They are source of oxalates. Source of oxalate. What is the source of oxalate? Ascorbic and glycine, because ascorbic acid is vitamin C and glycine. No other amino acid gives rise to oxalic acid. Okay. Serine, when decarboxylated, will produce ethanolamine. Glutathione is used in body for all the following functions except glutathione is used for all except methemoglobin is nothing but oxidized hemoglobin. For ammonia trapping, glutathione, there is no role of glutathione is in ammonia trapping. Okay, glycine for benzoate is used for ammonia trapping or for removal of glycine, but the answer here is for ammonia trapping. Reduction of methemoglobin to normal hemoglobin. Told you glutathione reduces or prevents the oxidation of hemoglobin. So oxidation of hemoglobin is methemoglobin, and it is converted to normal hemoglobin by glutathione, and it helps in carrying amino acids across membrane, and peroxidase, so it removes the peroxidase. Okay.